Okay, we're finishing up section 7 of chapter 2 because there's a lot of information here. Now we're referring to a clinical view for fatty acids, saturated, unsaturated, and trans fats. Most animal fats are saturated. They're solid at room temperature. Again, they have a flat, configuration and a single bond, whereas most vegetable fats are unsaturated. They're liquid at room temperature. They actually have that double bond, and they have more of a kinked structure, generally healthier. They converted to saturated fats through hydrogenation, so they can, but generally speaking, generally healthier. Partial hydrogenation may lead to trans fats, basically changing that cis configuration of an unsaturated fat to a trans configuration of a saturated fat. And then, of course, again, that trans fat ends up with the two most interesting parts of saturated and unsaturated fat. Ends up being rigid, like a saturated fat, and also ends up being kinked with that double bond structure, like unsaturated fat, making it hard to break down and utilize, and consequently use efficiently for energy. It's really the hardest for the body to make use of. So it ends up oftentimes becoming a high contributor to cholesterol. Carbohydrates with hydrogen and hydroxide are usually attached to every carbon. The chemical formula is CH2O, with N being the number of carbon atoms, all depending. Monosaccharides are simple sugar monomers. Disaccharides are formed from two monosaccharides, and polysaccharides are formed from many monosaccharides. Glucose is a 6-carbon carbohydrate, so C6, right? Most common of the monosaccharides. It's the primary nutrient supplying energy to the cells. Concentration must be carefully maintained. So the body wants to break that down when it wants energy, right? Most quickly. Glycogen is a liver and skeletal muscle stored glucose, essentially, and basically what happens is it binds glucose monomers together to form glycogenesis, and it breaks it down when it needs it, right? Hydrolyzes glycogen into glucose as needed, glycogenolysis. Liver can also form glucose from non-carb sources, gluconeogenesis. It takes up a lot of water. It's quite the strain on the liver, so it's really not ideal, but it can do that. The body would much rather have carbohydrate when it wants energy, or break down stored carbohydrate when it has it, when it's necessary. It would rather not try to use non-carbohydrate sources to form glucose, because those other sources that are non-carbohydrate have their own functions, and when they're used for the carbohydrate's job, because of the absence of carbohydrate, it really creates deficiencies within the system, and unfortunately people try to circumvent that on a regular basis with their nutritional advice sometimes given. Glucose and glycogen, basically, as you can see here, the glycogenesis, when you're taking that, we're trying to form complex carbohydrate, so to speak, that's stored, and then you're actually trying to break it down and use for glucose, glycogenolysis. Other types of carbohydrates: hexose monosaccharides, glucose isomers like lactose and fructose, 5-carbon monosaccharides or pentose sugars like ribose and deoxyribose, also used with nucleic acids, right, with DNA and RNA. Disaccharides are 2 simple sugars bonded together. The most common are sucrose, table sugar, lactose, milk sugar, and maltose, which is a malt sugar, all with glucose bonded to a second hexose monosaccharide. Other types of carbohydrates are polysaccharides, 3 or more sugars. It's really the most ideal carbohydrate basically to ingest when looking for energy for more sustained periods, as opposed to energy right now, which would be a monosaccharide. Glycogen is most common in animals. Starch and cellulose is found in plants. Plant starch is a major nutritional source of glucose for us as humans. Cellulose is a source of fiber. It's non-digestible, which is good if you're trying to pass things through. And if you have too much cellulose, it's going to create a lot of digestive challenges, because it's going to try to go right straight through you. Other simple carbohydrates, you can see here the monosaccharides, the 6-carbon sugars, hexose configuration with lactose and fructose, the 5-carbon sugars, pentose configuration with ribose and deoxyribose, disaccharides, sucrose, lactose, and maltose. So what's the repeating monomer of glycogen? Glucose monomers. Where is glycogen stored in the body? Liver and muscles. Where is it going to go first? Muscles, and then go to the liver. And when those are gone, your body has no more sustained glucose available. It's going to try to go to gluconeogenesis to actually convert other forms for carbohydrate, for glucose to break down for use for the body, and which means if you're trying to get that, your body's trying to do that, it's not going to give you energy right now. It's not going to have any for you. So consequently, you're going to have that, what some people call bonk, where the body no longer has any energy to perform anything that's of significant intensity beyond very basic life-sustaining type needs, like the ability to get up and move just a little bit. But if high-intensity type stuff, anything that's really tremendously challenging, it's going to need either straight glucose or stored glycogen used as energy, or it's just not going to have the capacity to perform work from a muscular standpoint. For each of the following, indicate if it is a monosaccharide, disaccharide, or polysaccharide: fructose, monosaccharide; galactose, monosaccharide; glucose, monosaccharide; glycogen, polysaccharide; lactose, disaccharide; maltose, disaccharide; starch, polysaccharide; and sucrose, disaccharide. With nucleic acids, nucleic acids store and transfer genetic information. There are two classes of nucleic acid. You've heard of these certainly: deoxyribonucleic acid, DNA, and ribonucleic acid, RNA. Both are polymers composed of nucleotide monomers, and monomers are linked covalently through phosphodiester bonds. Three components: the sugar, the 5-carbon pentose, okay, right over here. Then you have the phosphate group, which is attached to carbon 5 of the sugar. Okay, nitrogenous base over here attached to the same sugar at carbon 1 with a single or double ring structure. 5 types of nitrogenous bases are your three are pyrimidines, basically single ring bases: cytosine, uracil, and thymine. Two are purines, double ring bases: adenine and guanine. Nitrogenous bases within either group differ in functional groups attached to the ring. Now, in this case, the pyrimidines, you have your cytosine, your uracil, and your thymine. And basically, cytosine is both in DNA and RNA. Uracil is only in RNA. Thymine is only in DNA. And the purines for both DNA and RNA are adenine and guanine, side by side. So deoxyribonucleic acid, or DNA, is a double-stranded nucleic acid. It's located in your chromosomes, in the nucleus and the mitochondria. It includes basically a deoxyribose sugar, phosphate, and one of 4 nitrogenous bases: adenine, guanine, cytosine, and thymine. No uracil. Double strands are held together by hydrogen bonds. They form between the complementary bases. Thymine is paired with adenine, and guanine is paired with cytosine. Whereas ribonucleic acid, or RNA, is a single-stranded nucleic acid located in the nucleus and cytoplasm of the cell. Ribose sugar, phosphate, and one of 4 nitrogenous bases: adenine, guanine, cytosine, and uracil. No thymine. And so here we have the RNA single strand. We have the uracil unique to RNA. We have the nucleotide with a phosphate group, nitrogenous base, and ribose sugar. And we have adenine here and cytosine here. And you see the phosphodiester bonds showing there and there. This double helix DNA double strand, you see the hydrogen bonds will be attaching here between the nitrogenous bases, and of course unique to DNA is thymine. Nucleotide here with a nitrogenous base, deoxyribose sugar phosphate group, and this sugar phosphate backbone. Okay. Adenosine triphosphate, which we don't think of in terms of the DNA or an RNA, we think of in terms of energy. It's a nucleotide composed of nitrogenous base adenine, a ribose sugar, and 3 phosphate groups, triphosphate, right? It's the central molecule and transfer of chemical energy within the cell. It's necessary for muscular contraction. Without ATP, there is no muscular contraction, then there is no work performed. Covalent bonds between the last 2 phosphate groups are unique. They're energy rich. They release energy when they're broken, so that it basically ends up with from ATP down to ADP with an inorganic phosphate now floating around. And of course, ADP are going to try to get that inorganic phosphate to bind so they can again then break it apart for releasing energy for use by the cell for muscular work. I should say the cells. That said, it's an important nucleotide containing molecules that are going to be helping out here are the nicotinamide adenine dinucleotide, or NAD, as well as flavin adenine dinucleotide, or FAD, and both participate in production of ATP. So what's the general function of nucleic acids? Those nucleic acids are going to form, as you will note again here, break it down here for you, good old genetic information, right? DNA and RNA primarily. We don't typically think of that for the ATP, but certainly for DNA and RNA. And of course, the next question has to do with separating the differences between the structural differences between RNA and DNA. DNA has the basically double helix, the 2 strands. RNA has 1. RNA has ribose as part of the base, and DNA has deoxyribose. And basically, DNA uses thymine, and RNA uses uracil. Okay, and RNA is found basically in the cells in the nucleus of the cytoplasm and nucleus of the cell, and DNA is in the mitochondria in the nucleus of chromosomes. And we'll finish out with proteins for our final section here in just a bit.